Hi, I'm Brady Tickle.
Hi, I'm Michael Hall.
Hi, I'm James Aylward.
Hi, I'm Sam Tinnesz.
And our paper was Disruption of Astrocyte-Vascular Coupling in the Blood-Brain Barrier by Invading Glioma Cells.
As we've learned in class, astrocytes are glial cells that provide a link between the brain and the cardiovascular system, helping oxygen and other useful substances from the bloodstream get into the brain. They also maintain endothelial tight junctions that help make the blood-brain barrier largely impermeable. They accomplish this utilizing processes that form astrocytic infeed, covering roughly 99% of the cerebral vascular surface. These infeed also play an important role in regulating vascular tone via a process called neurovascular coupling, abbreviated NVC. Neuronal activity resulting in an increase of calcium ion concentration in astrocytic infeed also allows for the release of vasoactive molecules onto the smooth muscle cells lining the cerebral vasculature, which in turn causes dilation or constriction of these blood vessels based on neuronal energy demand.
The study at hand focused primarily on whether or not this astrocyte-vascular interaction could be disrupted by the presence of glial-derived brain tumors, or glioma, which are known to travel along the blood vessels before engulfing them and spreading with the help of angiogenesis, or the creation of new blood vessels following the apoptosis of one vessel. The basic idea was that as the glioma traveled in a paravascular fashion on top of the blood vessel, it would eventually get in between the astrocytic infeed and the smooth muscle cells lining the blood vessels. Keeping the function of the astrocytic infeed in mind, this would ultimately disrupt the astrocyte-regulated vasodilation and vasoconstriction of the blood vessels, as was the case in previous research using rodents. Thanks to Darwin, we can safely assume for experimentation that the same disruption could potentially happen in mice, and hence their brains were used in the current study.
In addition, another potentially disrupted function would be the degradation of the tight junctions of the blood-brain barrier, as the astrocytic infeed would no longer be connected to the smooth muscles and the vasculature. The degradation of these tight junctions would naturally result in easier access of blood-borne substances to the brain. If the disruption of the blood-brain barrier proves to be supported by experimentation, then there would be potentially fantastic implications for chemotherapy, as chemotherapeutic drugs could much more easily reach the brain and help fight against glioma if this barrier was made more permeable. For the results, in order to determine how glioma cells operate, the first primary experiment conducted involved injecting D54 eGFP human glioma cells, D54 glioma cells, into the mouse brain. As expected, the D54 cells traveled in a perivascular fashion along blood vessels. In addition, the D54 glioma cells also underwent co-option with the blood vessels, meaning they formed cuff-like clusters around the blood vessels. This way, when new vessels are created via angiogenesis, they end up feeding into D54 glioma cells, supplying nutrients that support tumor growth.
Along with observing the basic mechanics of how these glioma cells operate, the types of blood vessels that the glioma cells are attracted to most were also observed. Most of the injected D54 glioma cells associated with the capillaries that accounted for 47.5% of the total vessel area. About one-third of the glioma cells associated with penetrating arterioles or collecting veins, which accounted for 51% of the total vessel area. In addition, experimentation also showed a preference of D54 glioma cells for veins as opposed to arterioles. Finally, only a few cells associated with the large arteries and veins, which only accounted for 1.6 of the total vessel area. This indicated a preference of the D54 glioma cells for capillaries. Experimentation also determined that glioma cells do indeed displace astrocytic infeed from vasculature in order to gain further access to blood vessels. This was found using electron microscopy techniques analyzing the activities of D54 glioma cells, along with less important GBM22 and GBM14 cells in relation to astrocytic infeed.
In addition, the effects of vessel co-option on the blood-brain barrier were also observed. Using TRITC albumin and TRITC cadaverine dyes, it was found that the dyes were able to permeate the blood-brain barrier both at the site of the main tumor and at sites distant from where fewer glioma cells had spread. In vivo injection of Cascade Blue, a permeability marker, also found similar results, indicating a loss in tight junctions with the displaced astrocytic end feet. Glioma disruption was also found to interfere with neurovascular coupling of astrocytes and blood vessels. In a healthy brain, when astrocytic G protein coupled receptors are activated, there is an influx of calcium ions in the astrocyte and an activation of phospholipase A2, which converts membrane phospholipids into arachidonic acid. AA can either be released from the astrocyte and be converted to 20-HETE, which causes vasoconstriction, or be metabolized into PGE2, which causes vasodilation.
If the astrocyte-vascular coupling is disrupted by a glioma cell, the astrocyte loses control over the smooth muscles surrounding the blood vessels. This was illustrated via the application of the drug trans-ACPD and norepinephrine to acute brain slices. As these drugs increase calcium in astrocytes, in the healthy brain slices there was vasodilation and constriction of blood vessels. In glioma-afflicted brain slices, there was much less of a smooth muscle response. Finally, these results beg the question, can the infecting glioma cells control the constrictions and dilations of the overtaken smooth muscle of the blood vessels? Turns out this may very well be the case. Using the PAR1 G protein coupled receptor cells, which cause an increase in calcium concentration in glioma cells once activated, it was shown that glioma cells do get a say in the smooth muscle activities of their overtaken vessels. The activation of PAR1 by such molecules as thrombin and plasmin alters the configuration of potassium channels in glioma cells. When the potassium channels of glioma cells were blocked by inhibitors, it was found that the arterioles they were attached to constricted much less with PAR1 activation than arterioles whose glioma cells did not have the inhibition. This suggests that the glioma cells can play a role in vasoregulation.
In summary, the study found that invading glioma cells traveling in the perivascular fashion along the cerebral blood vessels do in fact disrupt astrocyte-vascular coupling, displacing the astrocytic end feet in such a way that degrades the tight junctions of the blood-brain barrier and prevents the astrocytes from controlling vasodilation and vasoconstriction. Ultimately, the engulfed blood vessels have the constriction and dilation of their surrounding smooth muscle cells controlled by the newly attached glioma cells, which use calcium-activated potassium channels to do so. While it was found that single glioma cell could potentially be enough to disrupt the blood-brain barrier, this finding does have hopeful indications for the treatment of this type of cancer. Oddly enough, the glioma cells, by working to disrupt the tight junctions, are opening themselves up to easier attack by chemotherapeutic drugs, giving these drugs much easier access to the brain with the degradation of the blood-brain barrier. However, as the study was conducted using earlier stages of the disease, early treatment is when chemotherapy would be deemed most efficient. Another major implication of this study is that there may be an advantage to glioma cells taking over dilation and constriction of the blood vessels. Invading glioma cells most likely constrict the blood vessels to have more room for perivascular migration, whereas growing satellite tumors could benefit from dilated vessels shunting additional blood towards the tumor. If this is the case, treatment of this cancer should focus on disrupting this control of the blood vessels by the glioma cells.
Finally, the last major implication of this study revolves around the loss of neurovascular coupling. This loss also results in the astrocytes no longer being able to provide energy metabolites from the blood to the brain, resulting in faster degradation of the brain. Impaired neurovascular coupling can also be seen in Alzheimer's, hypertension, and diabetes. Overall, this was a rather thorough study. However, it had one issue that we feel needs to be addressed. While observing this displacement of the astrocytic end feet by the D454 glioma cells via electron microscopy, the criteria used to distinguish the two cells was electron density. This is all well and good, but there's no real quantitative measurement being done. There is a higher electron density in the cytoplasm of a glioma cell as opposed to an astrocyte, for instance, and this was one of the factors that determined the difference. Unfortunately, there was no actual attempt to quantitatively compare the densities. Rather, the densities were just eyeballed under the microscope with no cutoff point of how dense the electrons had to be considered a glioma cell as opposed to an astrocyte. While this method was most likely fairly sound, there may have been some mistakes in identification without a true quantitative measure. Other than that, the study was very well done, and it definitely has great implications for cancer treatment and future research. In particular, it would be interesting to see the effectiveness of chemotherapy early on in the development of cerebral glioma cells, knowing about its disruption of the blood-brain barrier.